All right, so the special senses is fascinating topic. We will discuss, mostly the vision, the taste, the hearing, and also the idea of the equilibrium. Okay, uh, as far as the touch goes, we already covered the touch. We discussed the touch receptor, mechanoreceptor, proprioceptor. Those things already in chapter 13. So as far as the chemical senses goes, there are chemoreceptors involved, obviously. So the chemoreceptors are involved. They detect chemicals in the environment. Okay, everything must be dissolved. So when we're tasting something, like let's say something is salty, the salt should get dissolved in the saliva. Same thing with the smell. The chemoreceptor works best when there are detecting chemicals in the aqueous environment. Okay, so the smell is the same thing. All right, the substances must dissolve in the fluid of the nasal membrane, whether it's your favorite aftershave or perfume or the smell of the flower or the food. All of those is enters the nasal cavity, and in there are a lot of mucus lining, which you learn in anatomy. They get dissolved in there, and then you have those olfactory receptors waiting, which again, if you reviewed earlier on chapter, I believe was chapter 11 at the end, or 13, I can't recall quite well, but it's a G-link protein that is involved in the sense of the smell. So we'll take time and go over it really quickly and give you guys idea of what's going on. So as far as the taste goes, again, in anatomy, you learn that there are a lot of papillas on the surface of the tongue, and and also there are a lot of taste buds. So we go ahead and take a look at a small section right over here. These are called papilla. Basically, they're finger-like projections. And on the side right over here, you could see the taste buds. All right, the taste buds have been enlarged right over here. So one of these have been enlarged over here, so you could see it. And what do we see? We see gustatory hairs, which is another name for, the kind of like a part of a receptor. So you have gustatory receptor cells, which is our chemoreceptors. And there are other supporting cells around them. These supporting cells help out in terms of their structural ability and construction, as well as the stability of the cells. They're putting whole things together. Notice that there are, taste fibers. These taste fibers obviously are part of the cranial nerve. In other words, taking information. We discussed this already. Sensory neuron take information to the control center. In this case, would be the brain. All right. So these are that involved. So there are a lot of chemicals in our food. These chemicals in our food, which happen to be in the oral cavity, enters the taste pores. Then these taste chemoreceptors detect them, and then they send in their neurotransmitter and they communicate with these nerve cells. And these nerve cells are, again, are sensory neurons. Remember, we had receptors, which are this part right over here. Then you had the sensory neuron, which is this part right over here. But there are a lot of them that kind of like collectively, they're going in towards the brain, which happen to be the control center. So first of all, this part right over here is the aqueous environment right here. That would be your saliva. Okay, so that's what we're going to get at. We go ahead and take a look at the different types of taste sensation. Although this is very, very general, a lot more have been identified, but we're not going to get into it. Sweet, bitter, umami, those are all work via the G-link protein. Okay, so again, the others like umami, that kind of like beefy taste, or I don't know if you ever had umami burger and whatnot. They have a unique taste. It's not really salty. It's not really bitter. It's not None of the above. So Japanese scientists discovered this, and they are elicited by the amino acid glutamate. As you know, the glutamate was one of those amino acids that was involved in the EPSP, excitatory postsynaptic potential. Anyways, so all of these, they're unique in a way that they have outside receptors, molecules or the chemical bind to the receptors. These receptors are linked to the G-link protein, and as a result of that, the G-link protein can, you know, turn on a lot of relay molecules that we discussed in a previous lecture, including adenylate cyclase, hydrolysis of ADP to the cyclic AMP, and again, cyclic AMP can open up certain channels, and as a result of that, ions can go in and out. All right. However, the salt and the sour, they're really related to the cation. As far as the sour goes, that would be the hydrogen ion. As far as the salt goes, as you know, sodium chloride, they have ionic bonds between them. And again, when they are separated, they become Na plus and Cl minus. And this Na plus, due to the channels that exist there, can enter and cause the depolarization and therefore triggering the action potential as you're familiar with. All right, so let's get to it really quickly. There are proposed mechanism. So again, they must dissolve in the saliva. They must get picked up by the receptors. And after that, it's just the idea is the depolarization and releasing of the neurotransmitter. Neurotransmitter communicate with other neurons. Other neuron triggers their action potential, and as a result of that, that action potential is going to reach the brain, and that's what is happening. So that's the idea between pretty much uh, most of these chemoreceptors that are involved. Now, how they generate action potential and release the neurotransmitter varies. They could be either due to the sodium and calcium or hydrogen ion, or they could be due to the G-link protein that we discussed. Let's go ahead and take a look. Uh, in a nutshell, that's what's going to be happening. And information from the various part of our tongue where the receptors are located, we'll take that information and we'll take it to the side of the particular part of the brain that you studied in anatomy, gustatory center, and then you'll be able to determine or interpret as this signal what is the interpretation of this signal. Is it sweet? Is it salty? Whatever the case might be. All right. So again, you learn a lot of different type of cranial nerve that are responsible for taste, movement of the tongues, things in that nature. I'm not going to go over those right now because you're supposed to have that already. So let's get to the travel inside of the tongue and take a look at the receptor cells. So earlier I told you that these yellow ones are the receptor cells right over here. All right, you could see it. So if we go ahead and take this and kind of like show it to you, it'll look something like this. All right, pretend this is a yellow one. Okay, and again, if I upright it, then I could just draw it this way to make it simple. All right, these are the same thing. All right, these are this is the same thing as that one. This is the same thing as these yellow one, and there happen to be, receptor cells. So if we go ahead and just take a look at one of these on the next page, all right, perhaps, again, you could do this at home if you like to, you could map to see which type of receptors you have where. So for example, you don't need to know for the exam or the quizzes, but the tip of the tongue, you could detect the sweet better, for example, all right, and salty. On the side of the tongue, you could detect the sour and the bitter. you could detect towards the back of the tongue. So again, if you like to do these experiments, you could have a family member, for example, have your eyes closed and put in sugar molecule or salt or different type of chemicals in your tongue while your tongue is sticking out, all right, one at a time. And then you bring your tongue inside and you try to use your saliva to see if you could tell what it is, all right? So again, you kind of like could map these uh, and you will find out the location in which those chemicals or substances are placed on your tongue varies. And some location you could detect it right away. All right. You put a drop of vinegar, for example, on the side of your tongue, you could probably tell right away versus the other one. Make sure if you do that kind of experiment at home for a fun purposes, uh, to rinse your mouth with water every time you're doing this. Okay, so that's another criteria you could do. But let's go ahead and take a look at one of these receptor cells to see what it looks like. In this particular case, I wanted to show you guys because I've already done it. So this is what the salty tastes like. So sodium chloride in the saliva or water aqueous environment, they dissociate into sodium cation and the chloride anion. So this sodium cation is the important part. So there are protein channels just for sodium. So when they get dissociated, guess what? They can go right in. And because it's a cation, this could change the voltage of inside of the receptor cells. And that caused depolarization. This depolarization further down is going to open up various voltage gated channels, just like the neurons that we discussed. And it's going to reach towards the end where the calcium voltage sensitive channels can open and the calcium can come in. Now, this calcium is coming in, causing the exiting or exocytosis of the vesicles with the neurotransmitter, most likely glutamate. And this glutamate or the neurotransmitter that are releasing, again, bind to the ligand or chemical gated channel on the nearby neuron, which again, they trigger the action potential because it's excitatory, and then it sends a signal to the central nervous system. What if action potential did not fire? Then you wouldn't be able to taste uh, that salt. There could be lower amount of salt in there, and as a result of that, lower amount of neurotransmitter being released, and as a result of that, that may not be enough to reach the threshold on these nerves. So these nerves are the sensory nerves, sensory neuron. All right. So earlier I told you in this diagram, these are the sensory neuron. So the cells that are in yellow, there happen to be the chemoreceptors that we discussed. And I just showed it to you separately. All right. So again, we have all these receptors all over your tongue. What about the other ones? Let's go ahead and take a look at the next one, which happened to be a detection of uh, sour materials, say vinegar or something that is sour, sour cherry, sour, whatever. Okay, so most of the time is a hydrochloric acid that they dissociate into hydrogen ion and chloride ion. The concept is very much the same. Hydrogen ion channels are located on these chemoreceptors, and as a result of that, they could go right in, all right, through their channel. We discussed this was the facilitative diffusion. So when you have higher amount of hydrogen ion outside, they're going to go right in. Inside, they are negatively charged. They are cells. These are happen to be the chemoreceptors. So as the hydrogen ion comes in, that could reach the threshold right around negative 55 that we discussed. Action potential is going to trigger due to this depolarization. These action potential voltage differences is going to reach the calcium voltage gated channel, calcium moving in and causing the exocytosis of the neurotransmitter. Again, neurotransmitter communicate with the sensory neuron, which again communicate with the central nervous system or the brain, and the brain is the control center and it can interpret things. So sweet and sour are really simple. They just have their ion channels that they could go right in, and as a result of that, depolarization can achieve. What about the rest? The rest, whether it's sweet, whether it's bitter or umami or even other ones, now they're talking about other molecules and they are investigating it currently. They're all triggered by uh, the G-link protein. The mechanism of it, there are several different proposed mechanism of it, but in general, I'm showing you what was talked about in your book. Again, if you read some certain articles and so on, there might be a slightly different according to different findings. But here it is the sugar or the sweet sensation when you're tasting a sugar. Sugar is a glucose, so this is a glucose, for example. The glucose, they don't just go in. First of all, they're too large compared to the cation. All right. Another thing is they do not have the receptors in order to their protein transport protein to get inside. So they bind to the receptor, and you could see these receptors are located right over here. So once they bind to the receptor of these cells, G protein gets activated. G protein can activate adenylate cyclase. All right, AD for adenylate cyclase, which again, it can trigger hydrolysis of ATP. This is an enzyme that can break down the ATP to cyclic AMP. Cyclic AMP is the second messenger. In this case, this second messenger closing the potassium kind of like leak channels, so potassium no longer going out, and that causing the depolarization, which again, this depolarization, when it's great enough, is going to cause the action potential to fire, and the rest of the story is the same. All right, so just know that there is a G-link protein is involved. The cascade of the G-link protein, you should familiar with it as well. The mechanism over here, or proposed mechanism, there could be other mechanisms involved too. So just letting you know. Next up, we got the bitter taste. Bitter taste usually because of the different molecule. Quinine is one of those. If you have some quinine, you'll taste the bitter taste. Certain alcohol, not only they have sweet, but also they have a bitter taste to them. Certain beers, for example, and whatnot. But anyways, as the molecule dissolve in the saliva, they find themselves to their specific receptor. Again, this is the same idea: G-link protein, adenylate cyclase, cyclic AMP. And finally, this cyclic AMP can do different things. In this case, it can cause the smooth endoplasmic reticulum to release the calcium, which is sufficient enough to cause the exocytosis of these neurotransmitter. The rest of the story is the same. So in a nutshell, everything is involved with G-link protein except the salty, which is the sodium ion is involved, and the sour taste, which is the hydrogen ion is involved. All right. So that's as far as the taste I would like to talk about. We go straight to the olfactory senses or sense of smell.
Professor,
Let me pause the video, see if you guys have a question, and then we can go ahead and talk a little bit about your question. Hold on 1 second. All right, next up, we want to go over the olfactory senses. Olfactory senses are really important. So as you know, taste is 80% smell, all right? If for whatever reason you cannot smell, food that you eat become very tasteless. People like, I'm sure you have as well, you caught a cold at one point in time, and you can't smell things and your appetite is being suppressed. You don't feel like eating anything or whatnot, and anything you eat, it just doesn't taste the same. All right. Uh, my advice to you, never take your friends out anywhere or invite them over if they are having a nasal congestion, because they cannot appreciate the food that you prepare for them or whatnot. Okay. Uh, Nowadays with the COVID-19, is obviously, there's not many people gathering to begin with. But interestingly enough, and maybe those people that are presenting about the COVID-19, maybe you could talk about why that the sense of a smell of certain patient disappears during that. Okay, but in general, there's a lot of mucus lining that they starts producing more and more mucus. That's why you get the congestion on the nose and so on. So preventing you from picking up molecules readily, and as a result of that, you can't smell well. All right. So that's the idea behind it. But a lot of things influence our taste, including thermoreceptors, which is again the ability of substances getting dissolved. It plays an important role. Having your ice cream while it's completely melted, it will taste very sweet because there's a lot of sugar now that are dissolved and so on. Mechanoreceptor, how you feel with your touch receptors on the surface of your tongue. You have nociceptors. These are for pain. Again, something that is hot or is spicy, or perhaps could create painful stimulation. So temperature, they're all playing an important role, obviously, when it comes to the uh, you know, taste. So we go ahead and take a look at the sense of smell. As you know, the sense of a smell, molecules, they travel inside of the nasal cavity. Right over here, this rectangular area have been enlarged in here, so you could see the mucus lining, all right, the mucus layer. So these molecules get dissolved right over here. And again, you could see the olfactory receptor cells right over here, right? They detect these. So these are also known as chemoreceptors. When we say chemoreceptors, it's just general terminology that they can detect chemicals in the fluid. All right, so right over here, they can detect these. Let's attach them to them and so on. And again, same ideas. By the G-link mechanism, the action potential is going to get triggered and is going to propagate along this, and again is going to communicate with other nerves, and these nerves obviously it's goes into the olfactory tract and part of the brain that is responsible for sensing the smell. Interestingly enough, our sense of a smell, it looks like through evolutionary course, have been declined. Why is that? I mean, when you're looking at some of the bone structure that we found on the early humanoid in the caves and so on, the nasal cavity or the hole for the nasal cavity seemed to be a lot larger than what it is today, which suggests that we had better and larger nose and better capability of sensing different chemicals. Of course, for the survival, it was necessary to, find potential mates, find perhaps maybe a food, and avoid predators. All of those was related for, and you use olfactory senses. But as the course of evolution took place, we became hunter and gatherers, and then pretty soon, humans were involved in trading and so on, and we seem to be using less and less of our olfactory senses. Don't get me wrong, it's still a lot of in psychology, a lot of scientists believe that the odor plays an important role in terms of the attraction and so on. But our ability to pick up these molecules have been reduced over time. All right. Dogs, for example, they are much keener, and many other organisms to sense of smell is a lot more enhanced than ours. Uh, I was watching some time ago how a predatory cat like tigers or lions they attack uh, the herds or animals and so on, and they always go opposite side of the wind because the wind can carry their smell, and the preys, they can sense that and get away, which I thought it was fascinating. That again tells you how sharp their sense of the smell is, and so on. Even sharks, they can smell the blood, for example, or other senses that they do have in the ocean. So there's really opening up a lot of different ways in which organisms use their olfactory senses. But let's get back to the ideas right over here. So what we're going to do, we're going to travel inside one of these olfactory receptor to see how the G-link protein is involved. And again, the idea is right over here. So this is olfactory receptor cells. All right, that's the membrane right over here, and this is the outside of the olfactory cells. All right, so this is the inside. All right. So as you can see, the odor chemicals are coming in and binding to the receptors. All right. Once it binds to the receptor, now it's bind to the receptor. So a little bit later time, this activates the G-link protein, which finally activates the adenylate cyclase, which finally, hydrolyzes the ATTV to the cyclic AMP. In this case, cyclic AMP opens up the sodium and the calcium channel, as you can see in here. Sodium for the red, for the yellow ones, and the calcium maybe we can put in red or we can put in a black one. Doesn't matter really. But both of these can go, and once the channels are open, they can just go right in, and as they're going right in, this is positive ions, so it could cause the depolarization, and again triggers the action potential with propagation and so on that we discussed. The action potential can propagate through these receptors and then again release the neurotransmitters right around here, which again it can bind to the ligand gated channels, and then again communicate with other nerves that are going towards the olfactory tract and towards the brain. Okay, so pretty simple when you think about it. It's just G-link protein is involved, and you're familiar with it. This is just major mechanism involved. All right, so we're done with the taste, we're done with the olfactory. Let's go ahead and take a look at the eye. When it comes to the eye, our eyes are actually pretty good. 70% of all sensory receptors happen to be located in the eye. That means what? That means we use our eyes a lot for doing different tasks. Right now, you're using your eyes to looking at the screen and also listening. We'll talk about the sense of hearing later, but your eyes really, really important to bring in a lot of information from outside environment. Okay, there are a lot of accessory structure. These accessory structure help out. These includes the eyebrows, for example, that can provide some kind of a shade, eyelids that can distribute the fluid so your eyes doesn't get dry, also protect your eyes against the pollution, dust particles, things in that nature. Conjunctiva is a connective tissue, a layer that prevent microbes to enter and cause problem. Lacrimal apparatus that produces tears, again constantly washes and moisten your eyes so there won't be any irritation, and if there is anything there it could help to bring it out. And then also extrinsic eye muscles that you learn in anatomy enables you to move your eyes in different directions left, right, up, depression, and you know, elevation. All of those were the actions that you learned in the extrinsic eye muscle. Moving forward, we're going over in general, there were three layers of the eye. One of the layers on the outside was the fibrous tonic, which again was a sclera and connective tissue. The middle part was the vascular tonic, which is mostly choroid, blood vessels, and so on. And then the inner layer was the sensory tonic, and that would be the place where the photoreceptors were located. Those were all in anatomy. Nearby, there was a pigmented layer. It's important to know your anatomy, so review those if you can. And then once we get to the pigmented layer, you could see a lot of neural layers. There are nerves that are communicating with one another. Again, in anatomy, you learn that the photoreceptors were the rods and cones, but there were also bipolar cells. There were also other cells known as horizontal and amacrine cells. Let's go ahead and look at it. So we're looking at the light as the light passes the lens and going towards the back or the surrounding or first layer of the eye, which is called retina. You could see the pigmented layer. Pigmented layer as they're made out of melanin. Melanin is a protein molecule that is darker, kind of like gives a color to the skin. And if you're wearing dark color and you're going outside, and today is pretty sunny, it absorbs all the light, so you get pretty hot if you're wearing dark clothes. Most of the people wearing lighter color in the hot days, like white t-shirt or pastel colors, things in that nature. So it doesn't absorbs the light. Same thing in here. This pigmented layer absorbs the light. So as the light coming in, they get absorbed in the pigmented layer. Guess what? Nearby, there are rods and cones. We have more rods. Rods are responsible for distinguishing, very much seeing dim lights at nighttime, specifically. Rods are becoming really handy. You could pick up movement. These are peripheral vision related to the rods, and seeing different shade of uh, black and white, for example, gray color, things in that nature. Those are all the responsibility of the rods. We do, however, have cones. Majority of the cones are concentrated in the area that you learned in anatomy, and that would be fovea centralis, um, and where the macula is. And again, you might have heard about macular degeneration when you have a lot of damages to the cones, and as a result of that, you cannot see clear vision or central vision. Cones are starting with C, so the way that you could associate with this, clear vision starts with C as well, clear. Color vision, C. The genetic problems that I gave you guys related to the color blindness and so on, those are all the cones that are involved or malfunction. And also central vision. So they're triple C, central, clear, and color. Okay, so those are really essential as well. And by the way, make sure that you review the genetic, especially when it comes to the blood, whether it was a sickle cell, whether it was, blood typing, uh, whether it was the genetics of the blood. Certain people with certain blood types, they are, let's say, reproducing. Make sure that you review the genetic parts as well. But you could also look at the guidelines. Anyway, so what's happening in here? Again, once the light picked up by the photoreceptors, they're releasing their neurotransmitters right here, and the neurotransmitter communicating with bipolar cells, which you could see it in the red color right over here. And again, other cells, specifically a macron and the ganglion cells. And the ganglion cells, you could see they're kind of like bundling with one another, which together they go back of the eye, and that was bundle. All these bundles are known as the optic nerve. All right, optic nerve is basically the extension of all these axons of the ganglion cells, all right, collectively. And they exit through the eye, and again they go to the occipital lobe, where we have a visual association area, and you could determine whether you see color, whether you see movement, whether you see, peripheral vision, things in that nature. All right, moving right along, if we go ahead and take a look at the photoreceptors, they're located everywhere. All these areas, they have photoreceptors. Again, think about three-dimensionally, except this part right over here. This part is called the optic disc or blind spot. We do not have any photoreceptors in here because this is for the passage of blood vessels and the nerves. So if the object falls in here, then you will not be able to see it. All right. So, however, we don't really feel it because the blind spot of one eye is covered by the visual field of the other eye. But if you had one eye to operate and so on, there will be some areas in which you may not be able to see, especially if the images fall in that direction. All right, so there are different other parts to it. We already went over the jobs of the rods and the cones. So I'm just going to close this and go to the next one. And that would be part 2. The photo transduction, it's rather complicated process. And again, you might be able to look at some of the YouTube video to get the better grasp of it. I'm just going to make it concise, informative, and yet easy to understand. All right. So inside of the rods and the cones, there are kind of like a coin-like structures. All right. These coin-like structures, they have plasma membrane that can change shape as they absorb light, and awful lot of things happening in those areas as well. So let's go ahead and take a look at it. So we are traveling inside of the, let's say, rods in this case, and inside of these, there are tiny little coin-like structures right over here. All right. And again, if you look at one of these and enlarge it, you could see the membrane in there, and you could see a special protein as well. Now, these proteins are known as opsin, but whenever another molecule bind to it, called retinol, then together they're known as rhodopsin. So you got opsin. When the retinol is not bind to it, it looks like retinol already bind to this one. So this is called a rhodopsin. What's the big deal? Well, here's what happens when it's dark, all right, and when it's light. Two things happening, all right? They're different. So when it's dark, this opsin and retinol pretty much connected together, all right? So you have rhodopsin. However, when the light comes in, it changes the conformation of the retinol and no longer have the same shape. So that changing shape is going to cause a lot of changes. Let's go ahead and take a look at what happens, and then you guys can read the lecture on your own, as well as looking at the YouTube videos that I provided for you. So here is the retinol. Retinol is just a molecule. This retinol you could actually synthesize from vitamin A. So the carrot has good vitamin A's. It's a good idea to incorporate carrot in your diet to get these as well. Now, this is called the rhodopsin. It's just two molecules joining together. And now there is no light coming in. We are in complete dark. But what happens if you go to a room and you turn on the lights? Switching the light is going to cause this Remember, where are we at this point in time? We are happen to be in the coin-like structures. The coin-like structure happen to be on the rods or the cones. So those are the photoreceptors. Photoreceptors happen to be near the pigmented layer. So when you turn on the light, it absorbs that light towards the pigmented areas. All right. Here is the pigmented area with the melanin that we discussed that has the darker areas, and they are right in the vicinity. So now this cone-like structure with rhodopsin, they can absorb the light, and as a result of that, it changes the shape of retinal. Bear with me, I know it could get a little bit complicated, but if you review it a couple of times, it's not as bad actually. So now the retinol and the opsin are separated from one another. What now? This opsin is kind of, not exactly, but it's kind of like a G-link protein. In other words, they are involved in a cascades of events. All right. There's a cascades of events taking place. All right. Which result in voltage differences, not necessarily positive or depolarization, but voltage differences. In this case, hyperpolarization. Let's go ahead and take a look at what is happening. So light comes in, the retinal changes conformation. Now is more different looking. In this case, it was bent, but now it's no longer bent. So this activates the opsin. Now the opsin is activated, kind of like a G-link protein. It activates another molecule. This molecule is called transducin, which activates another molecule known as phosphodiesterase, PDE, which is an enzyme, kind of like adenylate cyclase. And what does this enzyme, phosphodiesterase, do? Phosphodiesterase detach the cyclic GMP. Now, cyclic GMP, they are always attached to the certain sodium ion channels. All right, so a little bit, not a whole lot, little bit of sodium continuously coming in. That is during the dark era. When the light comes in and the cascades of event taking place, it remove and disattach the cyclic GMP, and then what happens? Sodium channel closes. Sodium can no longer go in, and so normally we discuss inside of the nerve was a negative 70 millivolts, but during the dark time, because the sodium keeps coming in a little bit at a time, we are looking at our resting potential at the negative 40. This is the reason why, because the sodium a little bit at a time, it keeps coming in. Think of it as a leak channel, but these leak channel dependent on this molecule, all right? Once it's attached to it, or think of it as a fancy hotel, they have a doorman or a nightclub or whatever, all right? When you coming in, somebody opens the door for you or keep the door open for, customers, all right? Holding on to the door so you could go in. But what happens when the light comes in is kind of like that doorman or that establishment is closed. No more anybody can come in, all right? So this is when this departs. When the doorman is gone, then nobody can enter. In this case, sodium ion cannot go in. And as a result of that, the negative 40 now becomes more hyperpolarized. Hyperpolarization causes, because no more sodium is going in. Remember, we're putting a voltage meter in here and we're reading the voltage. So earlier, we are looking at the negative 40 millivolts. Why? Because of the sodium is keep coming in. So when this one is read at the negative 40 millivolts. But when you turn on the light comes in and all these cascades of events taking place. And as a result of that, this channel closes, no longer sodium can come in. Sodium is positive. So in this case, they're going to become more negative. So more negative means it has to be more than negative 40. In this case, it looks like they're going in beyond even negative 70. This is due to the fact that the sodium no longer is coming in. So look, action potential was a voltage differences. In our traditional action potential, we went from negative 70 to positive 35, and there was a voltage differences which translated to a signal. Same thing in here, except that this is a hyperpolarization. It still is a voltage differences that can translate to a signal. How they communicate with the other nerves and what kind of a neurotransmitters and so on, that one is a little bit more complicated. I could explain it, but I will leave it to you guys. If you are interested to know, I will maybe open up another lecture to give you just for your own knowledge and so on, but I'm not going to ask you that because it's a little bit, it can get a little bit complex, and I'm not sure everybody in the class going to be on the same level to understand what's going on. All right, so for that reason, all you really need to know at this point in time, just like action potential, that the depolarization or voltage changes was the signal. In this case, hyperpolarization or changing the voltage from negative 40 to more hyperpolarization is a signal as well. And this signal obviously is important in sending to the brain, which the visual association area is taking place. So these are called phototransduction. If you like, watch a couple of videos to see how it's done. Again, there are useful videos for you guys to look at as well. All right. All right. All right. Let's go ahead and take a look at the rest of the lecture. As you know, there are three different type of cones, blue cones, green cones, and red cones. And having a functional cones is important because it gives us ability to see all the rainbow in different colors. Now, if one of these cones are malfunction or is not working properly, you may not be able to see certain colors. And again, there are different type of color blindness uh, in the laboratory, or perhaps maybe you could obtain that from the online. There is a Ishihara test. Ishihara test, you can look at to see if you can see different colors and so on. Color blindness occurs more in males. We discuss about the genetics of it. And take a look at all the wavelengths. Visible spectrum is just a small wavelength, somewhere from 380 to 740 or whatnot. All right. But before that, there is a UV light. We can't see UV light. There is an X-ray. We can't see X-ray. Gamma rays, microwaves, radio waves, infrared. So none of those we are able to see it by a naked eye. I'm going to open up a parenthesis. This is a problematic. Some of you guys that talking about the harmful substances, maybe you will talk about these. I don't know, but a lot of things we cannot see. There are tiny microwaves everywhere now with the cell phone and the 5Gs and all that. There are infrared, there are UV that is coming in from our solar system, from the sun. And again, um, you're supposed to, we're supposed to have the ozone layer to protect us against the UV light, but at times, there are depletion of the ozone layer, or still some UV light penetrates. So all of that is creating a problem. X-rays, sometimes you go to the doctor, they need to take X-rays, including dentist and so on. Again, getting yourself so much on these rays or the wavelength that we cannot observe might be dangerous and could cause a genetic problems. So like there could be mutation or ATGCs that we discussed. Those can change because of such substances can create instability in the molecules. Interestingly enough, when you wanted to travel something, you're going through the doors and they, kind of like look at you when, you want to do an MRI, when you do want to do a CAT scan, a lot of procedures uses those because they wanted to detect things, but that does not mean that it's healthy for you. Right? A lot of them are very, very dangerous and could cause cancer, right? Especially when you are exposed to it too much. Even the cell phone, I don't know if you're constantly on your cell phone talking and so on, that uh, microwaves near the brain. Some study suggests in rats that they put in a lot of microwaves around them. They all develop brain tumor and so on. So again, what you find in one organism may not be applicable to another, and sometimes cancer would take an enormous amount of time in order to be proven and so on. And the companies know that, and they try to get away with it. All right, and that is unfortunate, really, when you see certain substances are banned in Europe, but then it's legal here, or we can use it here, but somewhere else in the world is not, or vice versa. That gives you a red flag. All right, and we don't know much about it. More study needs to be done, and unfortunately, many companies using this type of excuses and so on, and they just try to push their product at any cost. All right. So may not be a bad idea to take a moment and take a look at various substances around us and educate us. Anytime you do not know what is safe or what is not safe, you could look it up and try to minimize it as much as possible. Not to be overly stressed over it, but try to minimize it. All right. And I'm sure some of you guys in your presentation might be talking about that, but we'll see. Your outline was not really core specific for me to know whether you are talking about it or not. But anyways, let's go ahead and take a look at the what's coming up next. So the refraction is basically when the light goes from one medium to another. Light bends. So if you have, for example, a beaker of water, the beaker of water, and you put in your spoon into it. Your spoon might feel like it's bending, but when you take it out, the spoon is straight. So how did that happen? Again, light goes from one medium to another, it bends. So a lot of times when the light reaching our focal point, or perhaps maybe rods, and then in particular mostly cones, in order to see it clearly and so on, it must bend in the right way. Okay, so this is called refraction. If you have a problem with the refraction, then it may not bend the right way, and it might, focal point might be more like before the fovea or after the fovea. So that creates problem. That's why there are corrected lenses for that. So let me go over the problem with the refraction. Here's the emmetropic eye. Emmetropic eye means normal vision. And you could see as the light coming in from different object, it bends as it goes to the cornea, as it goes to the lens, as it leaves the lens, and they all focus right on the fovea, which happen to be right around here. All right. In the myopic eye, this is a typical nearsightedness. Nearsightedness is basically, when they are the curvature of the eye is more elongated, all right, and the focus or the focal point is not on the fovea. It hasn't reached that. So notice like it's here. It's supposed to be here. So for that, concave lens is involved. Concave lens is another medium. So when the light hits this medium, it bends, but it bends in a way that it can converge and go and wind up in the fovea where we want it. This is the reason why when you go to optometrists, they will tell you, is one better or two better or three better? They keep changing the lens. And each of the lens depends on the number, has a certain concavity. And it helps you if you're seeing clearer and clearer and clearer, that means it's coming closer and closer and closer to the fovea. Same thing with the farsightedness, which is nowadays more people are seem to have more nearsightedness as opposed to the farsightedness. But the curvature of the eyes is shrinking in this case, and nobody really knows why that is. Some people say that it's genetic, some people say that it's also environmental, because when you are playing a lot of video games, you're looking at your phone and you read up, close up, everything is close, 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 close up. So then you develop a nearsightedness. Uh, if you be looking at the stars, if you be looking at somewhere far, if you, perhaps looking at the, binoculars, looking at all far distances and so on, maybe you would have developed that. But again, nobody really knows why the curvature changes, but you need a different lens, in this case, a convex lens. So again, when it goes to another medium, it bends in a way that is focused on the fovea. And this is called hyperopic or farsightedness. Then what is presbyopia, or perhaps uh, what happens if you have astigmatism? Astigmatism is when the cornea right over here is not shaped the way they are. They might be like, bending one way or swaying another way, or it could be kind of like, different curvature to it and so on. There are different type of astigmatism. All right, so that's as far as that goes. Then what about nearsightedness for elderly, presbyopia, or perhaps when people need reading glasses, something that I need to get one now, unfortunately. Right? I'm having a hard time sometimes, with my vision too. Uh, but last time I went to optometrist, they said, well, you could use one, but it's still, if you get closer to the object, you should be okay with it. If you don't want it to wear it, uh, because you have to wear it and then take it off and use the other one unless you have a bifocal lens. But the idea is that the lens is supposed to change shape. The lens is elastic, it changes shape, and as a result of that, you'll be able to focus close up or focus somewhere far. All right. So this is called accommodation. When we say accommodation, let me see if I have it somewhere. Accommodation. No, this is adaptation. Anyways, should be here somewhere. Let me just tell you what accommodation is, so that way you could figure out reading it. It is right here. Accommodation is lens is changing shape. All right. So that way, it's kind of like your automatic cameras. When you take your photo, your lens is automatically changing shape, so that way you could take perfect photos. In the old days, you had to play around with the distances, or you had to manually turn uh, the distance of the lens with one another, so you could get a better resolution. So you could go close up, you could go back, and so on, and that's what happened. This is called accommodation. All right. Unfortunately, when we grow older, our lens is incapable of doing that. So now you have no choice. If you wanted to look at the text on the book, for example, or the newspaper or whatever, unfortunately, you cannot. Lens is not doing its job, so you're playing around with the distance. You have to bring it closer to see it, or further to see it. All right, unless you have a magnifying glass, or perhaps for the other reason. Again, I apologize for the sound, you guys. I live next to the street, so if you're hearing car going by, sorry about that. Anyways, that's the idea behind it. Now, pupillary reflexes or constriction is also important, and we touched base by it in the sympathetic and parasympathetic. As you know, this is called the pupil. So at times the pupil can, uh, you know, uh, dilate, so you see bigger, and at times it could constrict, so the pupil would be smaller. So this is the eye, and your pupil would be smaller. So if you light a flashlight on a split second, not leave it on because too much light can damage your photoreceptors. But if you go to the mirror, turn on and off the flashlight, what you see is the pupillary, there is a pupillary reflexes. So it's the same idea. It senses the light, it sends the information to the brain via the motor neuron coming back, and again it causes the constriction of the thing. So basically it eliminates or it provides less light to get in, because too much light can be damaging your uh, photoreceptors. Versus at nighttime, there is not much light availability, so you want more light to go in. So this is the idea on the nighttime, and therefore you can bring in more light. Okay. Now, when you're wearing the sunglasses, your pupil is going to dilate because, again, you're mimicking the dark and so on. So make sure you have a UV light glasses that are legitimate, not counterfeit, because otherwise more UV can go in and cause damages. But if you have a UV light protector on your sunglasses, then it bounces back. Not many UV light can pass through. So that's the idea behind the vision. Adaptation is also important. For example, you get adapted. If I'm in the dark room and I'm getting adapted to this dark room, all of a sudden I go to the light, it takes a little bit of time for me to adjust. Similarly, if I'm on the hot sunny days, it's very bright, and I walk into a place like in a garage that have no light in it and it's really dark in there, what happened is I will not be able to see much. Maybe some of you guys experienced this when you were late in going to a movie theater on a sunny day. Uh, so you may not be able to find the seats right away. This is the reason why. Okay. So you need to have your eyes to get adjusted to the dark, and then you'll be able to see. So your rod's getting activated a little by little, and as a result of that, you can see. All right. So that was pretty much vision. Uh, we can go ahead and take a look at the hearing and the balance. And if you remember your anatomy, we have the outer ear, we have the middle ear, and we have the inner ear. The outer ear, again, I'm not going to go and talk too much about the properties of sound and so on, but they are pressure disturbance. And this pressure disturbance is usually shown as a sine wave. It's going to go right from the outer ear right to the tympanic membrane or the eardrum. The eardrum vibrates, and as a result of that, there are ossicles. These ossicles are important. They happen to be in the middle ear, malleus, incus, and stapes. You learn one by one, they start moving, vibrating, and amplifying the sound. All right. The very last one, which is the stapes right over here, is connected to the area known as the cochlea. It looks like a shell of the snail. All right. And there are some fluid in here. So as a result of this movement, the fluid is going to go on the run in here, and it's going to hit the apex, and again there are another chambers in there. It's going to go backwards and then hit the round window. But this fluid movement is going to cause what's in the organ of Corti, which happen to be the hair cells and tectorial membrane on the top, to move. And as a result of that movement, so it's the mechanoreceptors involved in hearing. Certain ions that can go in, particularly sodium or potassium or calcium, depends, but it causes the generation of the action potential with, again, communication with the other nerve, in this case, the cochlear nerve, taking the information to the central nervous system. Similarly, semicircular canal and the vestibule doing the similar thing in terms of the concept. The receptor's name is different, but the idea is very similar to the hair cells, and the idea is basically they detect the position of the head or the movement, rotation or angular or horizontal, and then sending the signal towards the central nervous system so you could balance yourself as well. All right, so more on that shortly. Um, so I kind of like went over this one, and I wanted you guys to take a look at YouTube video known as hearing transduction. Hearing transduction is important, and I think it's a good video that you guys can take a look at it because visually you can see it. And I'm not gonna play it at this time, but if you like to, you could look at the video that I had on the YouTube and so on, and it's showing you. But there are many other ones that you could look at. So if you get a chance, go ahead and take a look. But the idea is that from the outer ear, the ossicles move, and then when you get to the cochlea, that's what happens. There is a fluid on the scala vestibule that they go on the run. Okay, kind of like when you are throwing a rock on a stationary water. These fluid are known as a perilymph. Okay, so what is this structure? This structure is the cochlea that I unwinded so you could see it better. Okay, that's what it is. And a little bit later on, you should be able to see this again right over here. See, this is the cochlea that is unwind. So this is the stapes, and inside here, this arrow right here that I'm drawing on it is perilymph. So perilymph is going to go all the way to the edge right over here, the apex area, and then it's going to go all the way where the round window is, and then it's going to bounce back again. So fluid is going to move, just kind of like you're throwing a rock inside of a stagnant water, all right? But in the inside of the cochlear duct, cochlear duct, also known as a scala media. All right, there's also fluid in here that is known as endolymph. So they are moving as well, because surrounding this membrane right here is very, very thin. So when one is moving, the other one is moving. Think of it as a plastic bag that you have in the water, and then you throw in the rock. When you throw in the rock, the ripple effect of the water causing the plastic bag on the water to move as well. So because they are on each other, they are in the vicinity of each other. So what happens then? What happens then? I wanted to travel inside here, the cochlear duct. The cochlear duct is where where the organ of Corti is located. So if we go ahead and take a look at it, all right, inside here, you see this structure. This is the scala media or the cochlear duct. So the fluid is going to cause this tectorial membrane on top to move, which is right here. All right. And this is connected to what's known as a hair cells. So you could see these hair cells right over here, or also known as a stereocilia. They're going to move as well, because when this one is shifting, these hair cells, they're going to move. Movement of these hair cells, all right, is going to cause ions to move in and cause the depolarization, which later on is going to release the neurotransmitter glutamate, which later on is going to, communicate with the cochlear nerve that we discussed, which is happened to be right over here too. All right. So this part is easy. Let's go ahead and take a look at the hair cells, see what happens in here, and then we can go from there. So over here, we mentioned what is happening. Over here, there are hair cells, so movement of the endolymph causing the hair cells to bend. So over here, the hair cells are bending. You could see how the endolymph is moving. The arrow showing you the movement of it. tectorial movement or displacement occurs, and now you have the hair cells that are no longer like upright like that, all right, more like bended like this, all right, due to the movement of the liquid and the lymph. All right, so what does that do? Let's go ahead and take a look. It opens up a potassium channel and a calcium channel. Now, these two cations happen to be very rich inside of the endolymph. All right, so they're both cations. They can go in there and cause the action potential, and this action potential can propagate and release the neurotransmitter. And most likely, this neurotransmitter is glutamate. The rest of the story is the same. In other words, calcium goes in, releasing the exocytosis, neurotransmitter bind to the ligand-gated channels, more ions going in, possibly sodium and calcium, and causing depolarization, and the action potential is going to fire, and this information is going to reach the central nervous system. So again, if you look at the video, I think it's going to help you very well. There are some ideas about the properties of sound and the transmission of the sound. They're pretty straightforward if you wanted to go through it. But basically what is happening, movement of the ion, movement of the fluid is going to cause the tectorial membrane to move, and therefore the hair cells starts moving, and movement of these hair cells, they are unique. They are mechanoreceptors. Remember that. And as a result of that, ions, cations, they could go in and depolarize, and this causes the action potential and the communication via the many nerves, which again take the information to the auditory association area, which happen to be, a area that we could hear things. All right. And again, many of these structures, if it sounds unfamiliar to you, you're supposed to have it in the anatomy. If you haven't, you do need to review. I'm only going to give you guys a 10-point quiz on Monday related to stuff that I'm talking about right now. Okay, so moving right along, let's see what time it is. We have a 12:15. Okay, so let's go ahead and take a look at what about the vestibule. All right, if we go ahead and take a look at here, there is a vestibule right around here, and there is also semicircular canal attached to it. And I wanted to little bit, not a whole lot, elaborate on the vestibule and semicircular canal. All right, so let's go ahead and take a look at the vestibule to get the better idea what it does for us and what do we need to know about it. So these are equilibrium receptors, vestibule and semicircular canal. Both of them is important in terms of equilibrium. One is monitoring a static equilibrium versus semicircular canal monitoring dynamic equilibrium. What does it do? What does it mean by these? Think about, I'll give you a scenario to understand this, all right? And then hopefully I'll give you an example of each so you can, kind of like relate it. So this is maintaining orientation and balance in a space. Let's say you're jumping, and while you're jumping, you're rotating in the air, but then you land on your legs and you be fine. You could balance yourself. Astronauts, for instance, they need to balance themselves because you never know which direction they're going to go to. People that are in the boat, as they go up and down, as they go forward, there's a lot of balance involved in our everyday life. Even when you're jumping, even when you're running, balancing yourself is important. Otherwise, you're going to fall and probably hurt yourself. So for that reason, we're going to go ahead and take a look at these. Let's take a look. All right. I wanted to talk a little bit about the macula. These are the receptors for the static equilibrium, and they are a lot of supporting cells nearby, but most likely they are involved in detection of vertical and horizontal movement. All right. Again, what does that mean? Vertical and horizontal movement. Let's go take a look. Um, Right over here on the top, you could see the vestibule right over here. Okay, and there are two different parts to it. One is called the article, which is this portion that is I'm highlighting. Another part is, let's use a different color, the blue portion that I'm highlighting, and this is called the saccule. So if you look at the saccule, all right, the orientation of the hair cells are in this way. Okay, and you could see what it looks like on the orientation of the article on the red part is right here. In other words, they're more like upright. And take a look at these stereocilia or hair cells. They're kind of like an upright position. So one is in this shape, the other one is kind of like more upright. And there are fluid in here. You could see some of these, they're known as otoloid, but there are fluid in here as well. So if you go, let's say, on one of those ride in Magic Mountain, I don't even know if they're open or not, but let's say that you go to the free fall. There are a lot of fairs that used to have this free fall too. You go up and then you just come back down. So when you're coming back down, your whole system is going down. So this, I'm going to put the arrow, you're going downwards, like, you know, you're falling, all right? But the fluid is going to go upwards. So these hair cells is going to move in this direction. And as a result of that, again, you have a depolarization and release of neurotransmitter. And again, you have nerves right here, vestibular nerve, that communicate with that. And again, it gives you an impression that you're falling. Now, if you have your eyes closed, you might have experienced that. And if you're falling, you still feel like you're falling because it's not what you see a lot of times. Yes, vision can help too, but it's not what you see. It's basically uh, how this liquid is going to cause these cells or stereocellulars to move. And as a result of movement, same concept that we discussed, cations going in, causing depolarization, and as a result of that, action potential is going to fire. Well, what if you have your eyes closed, you're sitting on, let's say, inside of a train, and the train or the airplane is taking off super fast? Same thing. This part right over here, they're going to bend. All right, they're going to bend this way if you're going the opposite way. All right, because the fluid is going in the opposite way. Think about if you want to understand this. Pay attention. This is very easy to understand this. All right, pay attention to this one. All right, let's go back and get a new page. If you have a paintbrush, here's your paintbrush. When you brush down, in other words, when you holding in here, for example, with your fingers, all right, and you brushing downwards, the brush hairs goes upwards, right? So this is same concept in the vestibule. Versus, let's say that you're brushing the ceiling, you know, your ceiling is this way and you're brushing it and so on like that. All right, if you're moving forward, your the hair on the brush goes backwards, right? So it's kind of like that. But these are resembling the stereocilia. So when they are going backwards, what happens? There are channels in here that opens. These channels, when they are upright, they are closed. So no cation can go in. In here, the cation can come in and causing the depolarization. You know the rest of the story. And once you reach the end, there is a exocytosis of the neurotransmitter glutamate, and again communication with the other nerve to send the signal to the central nervous system. This is a similar concept. So when we say horizontal movement, or vertical movement, we're referring to as this. All right. But there is also a semicircular canal. And the semicircular canal, they are responding to the Christa Ampillaris is the name of it, to the angular movement. So now you're not going forward, you're not falling, you're not going upwards, you're not going backwards, all right? Angular movement. So now you have to think about rotation. How many different ways you could rotate? Think about it. You know, ice skater, for example, you know, they rotate, I don't know, they put their arms like that, and they rotate like that, okay? A diver, for example, I don't know if you've seen the divers, whatnot, but they, as they dive, they rotate several times like that. Okay. Uh, if you're doing cartwheel, if you're doing cartwheel, you know, you're rotating like this. They're all different way that you're rotating, right? All right. So depends on the type of a rotation that you do. Uh, we have different semicircular canal. And in here, I showed you three of them. And if you wanted to kind of like see it a little bit better, or maybe another diagram can show you, there are anterior, posterior, lateral semicircular canal. And again, there are fluid inside here. So it depends on which way you rotate. I'm going to give you an example of one for now. All right, so you get the feel for it. Let's go to the lateral one. Let's say that you're rotating this way, like an ice skater. So what happens, the fluid in here is going to rotate that way. All right. And again, the receptors are right over here. These are known as crista ampullaris. They detect that because the fluid that are rotating again is going to cause these kind of like a stereocilias, if the fluid is coming into bend. And as a result of that, same thing opens up the cation channel, cations goes in and causes a depolarization, and you feel like you're rotating. If you ever played that teacup, maybe in the fair, or simply when you're getting up, be careful, don't do that because I don't want you to fall and hurt yourself. But if you rotate several times like ice skated and then stop, what happens? You still feel like you're rotating. Well, why you feel like that is due to the fact that this fluid still is moving. When it stops moving, then you feel like, oh, okay, now I'm not moving. But even though when you're not moving, you feel like you're moving, you're rotating, and so on. That's the reason why. Now, when you're a diver, there could be fluid that is in this one, which happened to be the anterior one move. If you're doing a cartwheel, again, the fluid on the posterior can move, and so on. So I let you guys think about the three-dimensionally and the orientation and so on, but the idea is very much the same. All right, the receptor detects that, releases the chemical neurotransmitter. This chemical neurotransmitter, they go right into the vestibular nerve, and then again the vestibular nerve reaching the brain, and then you know how and which way you are rotating and such. Now, a lot of times, people getting seasick. They go into the ocean, for example, and they feel like vomiting, they feel like throwing up, and so on. Why is that? The reason behind that, or anytime you get motion sickness, is there is a miscommunication. Like your eyes is telling you one thing, and it seems like your vestibule and the semicircular canal might fire and tell you another thing. So when you're in the boat, you're going up and down. So your vestibule is detecting that. If there is a wind, you might actually rotate a little bit too. So a combination, some of your semicircular canal might kick in. But when you're looking at the horizon, you see a nice flat ocean, and you don't feel like going up and down or whatnot. So all of that, it may not settle to some individuals, especially if you don't have that much experience and so on in going to the ocean, or everybody is different. People sometimes they go or to the ride, which they rotate a lot and they find it thrilling and nothing happens to them. But another person going to the same ride might vomit and feel awful, and they never wanted to try that ever again. So again, just like any other receptors are sensitive to different experience and different uh, way in which they communicate, and therefore you might have a different response. It's not one response for all. Our human body is different. We are made out of different genes. We have different capabilities. All right, but motion sickness is thought to be miscommunication between the visual field as well as the vestibular and semicircular canal. So this summarizes what you needed to know for chapter 15. Let me go ahead and pause at this point in time and